Today we're starting on Chapter 5, which is going to be DNA, gene expression, and biotechnology. So what is it and what does it do? We're talking about DNA. So to start this out, let's discuss something that came about, the DNA 200. The DNA 200 are going to be 200 inmates who were released from prison because once DNA fingerprinting came about in the early 2000s, they re-looked at the evidence and they were able to analyze the DNA that was collected on the crime scene samples. And when they did that, they found that the DNA did not match those who were incarcerated. So what they did was they were able to go back, reanalyze this data, and release over 200 prisoners who were falsely imprisoned due to this mix-up. So what is the most common reason why DNA analyses overturn incorrect criminal convictions? Well, first you have to understand where that first criminal conviction came from. Most of the time, it is going to come from eyewitness testimony. So one of the most common reasons people were falsely accused was because somebody said, I saw this person do this, or they were able to pick this person out of a lineup and they accused them of whatever the crime was. So with the DNA 200, cases that were reopened where the DNA was analyzed, more than 3/4 of those cases have been inaccurate eyewitness testimony. And so here's some quotes that were made that are interesting. So selfish dictators may owe their behavior partly to their genes, according to a study that claims to have found a genetic link to ruthlessness. And that was done by a journal called Nature in April 2008. And then Time magazine goes on 2 years later to say, too many one-night stands? Blame your genes. According to a new study, it may be fair to say that while you jolly well couldn't help cheating, your particular genes did make things more difficult. And so now we refer to these times once we figured out how to decode the human genome. Now we call it the age of the genes. We start to see all of these different things come about, talking about what we've discovered. We can actually do DNA testing now. Lots of the famous ones are going to be like the one you see here on the right-hand corner, the Ancestry, or you can do a 23andMe. There's many other different DNA companies or DNA tests that you can have done. When you do these, you can not only find out your heritage or where you came from, it also can be plugged into specific apps. And with those apps, you can determine if you might have a gene for certain different types of allergies. You might have a gene for possible cancers. You might have a gene for different types of characteristics from brown hair, blue eyes, things such as that. And so with this, we've put a lot of research and a lot of time into studying what this DNA means. What can it determine? Down to determining if something is good or evil. They've actually found specific genes that might be more common in people who have what we consider to be evil tendencies or even serial killer type tendencies. So in summary, DNA is going to be a molecule that all living organisms carry in each and every cell of their body. Well, with exception to red blood cells. Unique in virtually every person, DNA can serve as an individual identifier left behind as we go about our lives. This is a fact that is used increasingly to ensure greater justice in our society, such as through establishing an innocence of individuals wrongly convicted of crimes. The DNA molecule is going to contain instructions for the development and functioning of all living organisms. So all living organisms will have a set of instructions that tells its body what to do and when to do it. All this started out with two important features in DNA. Beginning in the 1900s and continuing through the 1950s, two features were discovered that were going to be important. The first, DNA contains the instructions on how to create a body and control its growth and development. And two, these instructions are encoded in the DNA molecule and are passed down from parent to offspring. So scientists who worked on DNA, first you have the American Linus Pauling, who won a Nobel Prize in chemistry for his work and then elucidated the structure of these molecules and what they look like. Then you have Maurice Wilkins and Rosalind Franklin in England, which produce an X-ray picture of DNA that's going to be critical to coding its shape. It's referred to as Photo 51, and she used crystallography to look at that. Next, you have Francis Crick and James Watson. They were working in Cambridge, England, and they happened to put all these pieces together and deduce the exact structure of DNA. Now, they didn't come up with that structure on their own. They did use Rosalind Franklin's picture. They actually snuck into the lab and they viewed all of her work behind her back. And so that's how they finally came up with the correct structure for this. And here's a picture of Watson and Crick and their model that they created. So starting off with this DNA structure, first we refer to it as a double helix, which is going to be a twisted ladder shape. It's going to be made up of repeating nucleotides. Nucleotides are made up of a sugar, a phosphate, and a nitrogenous base. Each of these bases is going to have a base pair, and that's what it's going to match up with on the opposite side. And so bases from each side of the helix are going to come together in complementary pairs. They're going to be held together in the center by a weak hydrogen bond. So coming over here to this, we have this section of DNA. We're going to look at it over here larger. So anywhere you have an A, it's always going to match up with a T. If you have a T, it's going to match up with the A. Anywhere you have a C, it's going to match up with the G, or if you have a G, it matches up with the C. Now notice that they're going to run against each other. One is going to run one way, the other is going to run the exact opposite way. So that's why on this side, they look like they are upside down, because this strand is running here. This strand is going to be right side up because it is running this way. Right, next, looking more into that nucleotide structure, things that you have to know. First, you're going to have your sugar phosphate backbone. And we've already talked about this a little bit when we talked about macromolecules and we talked about the nucleotide. This should be a simple review, but you're always going to have a phosphate group on the side, and then you're going to have the sugar group it's attached to. These are going to be very strong bonds, and we know that strong bonds are going to be the covalent bonds. So these are very strong covalent bonds, and they're going to be referred to as the sides of the ladder. And so you're going to find it in this portion of DNA or in this portion of DNA that's kind of the greeny blue color. Very, very strong bonds. They do not separate like the center does. They're going to be responsible for keeping these DNA bases in order. You don't want them just randomly redistributing themselves or rearranging themselves. So this strong backbone of sugar phosphate is going to be responsible for keeping them in order. Next, you're going to have the nitrogen-containing base, or referred to as the nitrogenous base. This one is adenine, and so with it, it's always going to match up, remember, with the T or the thymine. And so adenine and thymine match up. You see here, it's going to have that bond. That's that weak hydrogen bond. So hydrogen bonds are going to be weak on purpose. That way they can unzip and zip back up very quickly. They're going to have to do this. They're unzipping in order to copy a specific gene or specific sequence that it needs to produce more of whatever substance or turn something on. It's going to zip itself back up as soon as it finishes copying. Or it might need to unzip itself to create a completely whole new strand right before that cell goes through mitosis. Right, so we see that double bond, and here we see another. And that one's going to be hydrogen bonds here. It's a triple bond between that section. And that's going to be between the guanine and cytosine. And so here you're going to see those weak hydrogen bonds can unzip and zip back up very seamlessly. Next, talking about the sugar, phosphate, and bases, like we said, if it is DNA, it's going to consist of A, T's, C's, and G's. Those are your base pairs. The sides of the ladder, we said, were made up of the sugar phosphate, very strong. The rungs, or the steps, or the center of the ladder, are going to be made up of these nitrogenous bases, and they're held together by those very weak hydrogen bonds. So here's a quick question. Which answer will base pair with the following sequence? And so we're trying to find a complementary. So we know A is going to go with T, G is going to go with C, TT, C would put A, and you see how we follow along. Which one of these matches up? If your answer was this one, then that will be correct. And that's going to be for DNA. Now, if I were to ask you to create an RNA, which we haven't talked about yet, RNA, remember, from macromolecules is going to use a U instead of the T. So if I ask for an RNA molecule, that would be that one. Okay, so in summary, DNA is going to be a nucleic acid. It's a macromolecule that stores information. It consists of individual units called nucleotides, which are going to be made up of a sugar, a phosphate, and a nitrogenous base. DNA structure is going to be resembling a twisted ladder, which we refer to as a double helix. This is going to have the sugar phosphates serving as the backbone, and then your base pairs are going to go across the center or make up the rungs of the ladder. Genes are going to be sections of DNA that contain instructions for making proteins. So, why is DNA considered the universal code of all life on Earth? Well, if you think about that, humans, all animals, all plants, even different types of fungi or bacteria, we're all going to be made up of the same nitrogenous base pairs and the same basic structure. It could be either in a DNA form, where it's going to be a double strand and a double helix, or it can be in an RNA form, where it's going to be a single strand. And with these strands, they could be either linear, like human DNA, or they can be in a circular form, like we find in bacteria or prokaryotes. So, everything from humans to an onion, a fruit fly, an amoeba, and even the eel, all contain the same bases to make up these organisms. So it's basically the same universal language. So all living things on Earth are made up of DNA or RNA. A, T, C, G, or U, if we're talking about RNA, are going to be the basic bases that we're going to pull from here. All are going to have the sugar phosphate backbone. All that are made up of DNA are going to be held together in the center by the weak hydrogen bond. So from genome to gene, looking here, you can see the cell. Inside the cell, you're going to have a nucleus, and inside that nucleus is where you're going to find the DNA. So the genome is going to be an organism's complete set of DNA. In eukaryotes, this is going to be found in every nucleus. If it's in a prokaryote, it's just floating around in the cytoplasm. And so here we are actually, since we have that nucleus, you're actually looking at a eukaryotic cell. Inside, you're going to have separate chromosomes. These chromosomes, or often referred to as chromatin when it's in its loose form like this, are going to consist of your linear strands of DNA. So one or more unique pieces of DNA. It's going to be circular in prokaryotes and linear in eukaryotes, and they come together to make up the organism's genome. So inside of a human, you're going to have 46 of these chromosomes inside of each nucleus, unless it's an egg or a sperm cell. Then it will only have 23 of these unique chromosomes inside of the nucleus. Inside the chromosomes, they're going to vary in length and in size, and they can consist of hundreds of millions of different base pairs. So inside of, let's say, a specific chromosome, you can go to a certain section, like you see highlighted here, and within that certain section, you can come to a gene. So a specific gene that's going to code for something. On average, these are going to be around 3,000 bases long, and they contain information necessary to produce all or part of a protein molecule. So let's say that for dessert, you want to have ice cream. Your body's going to have to make the protein lactase in order to break down the lactose that's in that milk. So it's going to go to the chromosome that makes that, find that specific gene on the chromosome, isolate that gene, and copy it. Once it copies it, it can make that protein. You can eat the ice cream and digest it just fine. So the number of chromosomes is going to vary amongst species. For example, corn has 10 unique chromosomes inside of the nucleus. Fruit flies only have 4 chromosomes. Dogs and chickens will have 39 different chromosomes. Goldfish have 47. Humans can have 46 chromosomes. Sometimes they can vary. If they vary, then you end up with different genetic disorders. And individuals with each of these species are going to inherit one copy of each chromosome from each parent. So looking at genes. Genes are a sequence of bases in DNA molecules that carry information necessary for producing a functional product. Usually that product, like we said, is going to be a protein molecule or RNA. It is found on a specific section of the chromosome. That section is called the locus. If you're talking about multiple sections, you would change that to loci. And so the locus, that's a section that you find that specific gene on a chromosome. And so here is showing you the specific gene is found at this locus, and then they enhanced it so you could see the gene section. Now with each gene or specific trait that you're looking for, it's going to be found on what's known as alleles. So our alternate versions of a gene that code for a same trait is basically the definition of an allele. You inherit one or more of a set from your mother and from your father. For example, there are some genes that are considered to be one gene or one allele traits, and then there's those that are going to be multi-located or multi-allele traits. For example, height. They think that there's going to be around 6 genes at different locations that code for height. You would get 3 from your mother, 3 from your father. So if you got 3 short alleles that you inherited from your mother, but 3 tall alleles that you inherited from your father, due to the type of genes they blend, you might end up being a medium height. Some traits, like we said, use one pair of alleles. That in those types of traits, you would get one from mom and one from dad. With a species, individuals sometimes have alternate versions of a gene for a given protein, and these instructions will result in different versions of that same trait. So looking here, we have an alternate version of a gene that codes for the same trait, certain protein production. And the protein is going to be influenced by the environment and, in some cases, other genes that produce the trait. So we're looking at color here in this flower. So you have allele 1 produces a certain protein, maybe under a certain circumstance, it's going to appear to be different colors of an orangey red. Allele 2 produces a specific protein. Under certain conditions, you're going to see white. And then allele 3 is going to produce a specific protein, and under certain conditions, it appears to be yellow. Now, these are 3 different versions that you could have for the exact same trait. The trait is going to be color in this case. So different people can have free or attached earlobes. The DNA that encodes for making free or attached earlobes is going to be called a blank. And there are going to be 2 different versions of it called a blank. Which one do you think? Yeah, you said number 4, then, sorry, you were correct. All right, so DNA is going to be a universal language that provides the instructions for building all structures of all living organisms. The full set of DNA an organism carries is called its genome. In prokaryotes, the DNA occurs in circular pieces. In eukaryotes, the genome is going to be divided among smaller linear strands of DNA called chromosomes. A gene is going to be a sequence of bases in a DNA molecule that carries the information necessary for producing a functional product, usually a protein molecule or RNA. So not all DNA is going to contain instructions for making proteins. So looking at genome size of various organisms, I'll let you glance all that. Notice how an onion is going to have more than 5 times as much DNA in every cell as a human does. And a newt and an amoeba are going to have even more. So you would think the more DNA, the more complex the organism. But it turns out that's not going to be so. So if an onion has 5 times as much DNA as a human, why doesn't it make them more complex? Well, looking at this, percentage of coding DNA that's actually found in various organisms compared to all the DNA. For humans, 2% is going to be actually coded for specific things. Fruit flies, 19%. Roundworms, 25%. And on to E. coli, which is 90%. So in with this coding, you have virtually all eukaryotic species. That amount of DNA present is going to be way far exceeding what they actually use to make specific things. And so they don't necessarily need all this DNA, you would think. The fact is, a huge proportion of base sequences in DNA, like we said, that doesn't code for anything, we have come to refer to it as junk DNA. So a lot of biologists are talking about this junk DNA. They thought, oh, it just separates the genes. And sometimes even within a gene, you're going to find junk in between that has to be taken out before it can actually copy and produce that protein or that gene. So what to do with all this junk? Why is it even there? So in humans, for example, we said genes make up less than 2% of the DNA. In many species, that proportion of DNA that codes for the gene is going to be even smaller. For example, with bacteria and viruses, they tend to have very little non-coding DNA, with genes making up about 90% or more of their DNA. In the eukaryotic cells, however, with exception to yeast, that we see an explosion here of non-coding DNA. It's going to be actually about 25% of what occurs within the genes and 75% that occurs between each gene. So even within the genes, you find this junk. And so they kept thinking, why is all this junk here? What's it do? What if we just took it all out and just used what we code for? Would that change things? First, they thought no. But then, the more that they looked at it, they realized that they think this junk DNA, or the DNA that we don't use to code for specific proteins, might actually have another function to it. And that function, they think, actually goes into making us unique. It makes our personalities. It's what makes us tick, they think. So looking here, coding versus non-coding DNA, you have specific regions. We have gene 1, which is all in purple. Notice how it's not all together. And gene 2 is in green. It's not all together. It's going to be separated with the different regions of non-coding DNA. And since it doesn't code for anything, we do refer to that as the junk. So in order to put the gene together, it's going to have the entire section. You're going to have what's referred to as introns and what's referred to as exons. And so when that transcription comes together, that's what's referred to when it's copying a gene inside the nucleus. It's going to eliminate the introns, or the blue. That's what we refer to as the junk DNA. So looking at this DNA here, you're going in, you're eliminating the introns, or the separation points, and you're taking out only the coding part to create your mRNA. And that's what's going to go to the ribosome to actually make the gene. So exons are kept and are what code for specific genes. Introns are the junk part that's going to be spliced and cut out, and then it's going to hook all of those exons together. So with the introns, we said they were the non-coding regions of DNA found between genes. They may take the form of short or long sequences that are repeated thousands and thousands of times. May also consist of gene fragments or duplicate versions of genes, and sometimes referred to what we know as pseudogenes, pseudo meaning false genes. And so since they're false genes, it just means they look like genes, but they really don't code for anything. And then recent scientists, like I said, think that this junk might actually be what makes you unique. So this is what's going to give you your personality or your unique characteristics. So only a small fraction of DNAs in eukaryotic species actually codes for genes. The function of the rest is still a mystery. How do genes work? Well, here's an overview. First, you have what's referred to as a genotype. All genes that are contained in an organism, and these types of alleles that make up an organism. Phenotype is going to be the physical manifestation of these instructions, basically what you see. So with a genotype, let's see, you could have a brown eye and a blue eye. And we will put that up there. So let's say brown eye is dominant, so we make a capital letter. Blue eye is recessive, so we make a lowercase letter. I have brown eyes, but my mother has blue, so I know I have a brown eye trait and I have a blue eye trait. Got the brown eye trait from my dad, and then the blue eye trait from my mom. So I know this is my genotype. These are the types of genes I'm made up of. Now, phenotype is what you see. So if my genotype is big B, little b, what you see is going to be this dominant trait, and that's going to be brown. And so brown is the phenotype. The genotype is the big B, little b, or the traits that I'm made up of. So how do genes work? Basically, in order to even get to the big B, little b, what has to happen is DNA inside of the nucleus, which you see is that purple thing in there, is going to be copied. So first, in order to copy it, it's going to have to untwist itself through this section here, and it's going to copy it kind of like a cookbook copying a cookie recipe there. And so this here is going to run along the strand of DNA, and it's going to create an mRNA strand, which is that yellow strand. Once that mRNA strand's created, it's going to go through the nucleoplasm to the nuclear pore of the nucleus, and it's going to go out of the nucleus. Once it's in the cytoplasm, which is all the light blue there, it's then going to come to a ribosome. The ribosome is going to be this blue structure here. It's actually in two pieces. One piece is the bottom piece, one piece is the top piece. Now, with this, these two pieces come together. The strand of mRNA is going to go through the center, and it's going to be read 3 at a time. When it reads them 3 at a time, they're called codons. So it's going to read the base pairs 3 at a time, like A, U, G is always going to be your start codon. It reads it. It takes a particle called a tRNA particle, which is this yellow particle here, goes out into the cytoplasm. It finds the amino acid that matches up with AUG, which is one of these little circles, brings it back to the ribosome, and it starts this protein chain. Once it starts the protein chain, it's going to keep bringing them back as it reads them 3 by 3 and hooking them together with polypeptide bonds. This creates your polypeptide chain. Once that chain's finished being made, then it can be released as that protein molecule. Now it has other things it has to go through first. Each one of these little dots, remember, is an amino acid. So you could put AA there if you want to for amino acid. Amino acids hooked together with polypeptide chains make that protein, but remember, then the protein's going to have to be corkscrewed or zigzagged. Then it's going to have to be bent. Then it's going to have to be attached to other protein molecules like this, or amino acid chains, and then it becomes a functional protein. It doesn't just work straight out as this amino acid chain here. It has to be structurally put together after that. So the process that happened inside of the nucleus, where we copied the mRNA strand, is referred to as transcription. What happened on the outside in the cytoplasm at the ribosome is called translation. So you copied the gene, transcription, you made the protein from the gene, translation. Moving on. All right. So the gene in strands of DNA are going to be a storehouse of information, like an instruction book or like a cookbook. The process by which the information is used to build an organism occurs in 2 steps. Transcription, where you copy the gene base sequence, and that's going to be in the nucleus, and then translation, where it takes that copy and it directs the production of a specific protein, and that's going to be at the ribosome in the cytoplasm. Building organisms through gene expression, information in DNA is going to direct the production of these molecules that make up an organism. So we're going to pull all this together basically in our recipe and end up with a product. Here they're showing you cookies. So the transcription is going to be reading the information in that code from DNA. So step 1, it's going to have to recognize, bind, and unwind. And so it recognizes where that gene starts. So the enzyme RNA polymerase is going to go in. The space where it starts is called the promoter site. Basically, it's saying start here. The molecule is going to bind to one strand of DNA and it's going to begin to read that gene's messages. And so it's going to run along that line. As it's reading it, it's unwinding the DNA molecule just a little bit. So only one strand of that DNA can be read at a time. So it's reading it and it's matching it up with nucleotides and making a complementary strand doing that. As it reads and makes a strand, it's going to twist back those sections that it just read. So see, it's going to be twisting it back. Meanwhile, it's reading along here and untwisting here. So it's not going to untwist all of it and then go back and twist it back later. It untwists as it reads and then twists it back once it's finished reading that little section. Step 2 is then going to transcribe. So as the DNA strand is processed through the RNA polymerase is going to build the copy, and that copy of the gene is referred to as the transcript. And so now that mRNA strand is referred to as transcript. This copy also can be referred to as the mRNA or messenger RNA. So the transcript and the messenger RNA, same thing. They exit that nucleus. Step 3, rewind. As the mRNA polymerase is moving down that strand, copying, like I said, it is rewinding behind it. And so it unwinds, copies, and rewinds and twists it back into its normal structure. Step 4, it's going to come to a gene that says stop here, and that's referred to as the termination sequence. There, it's going to detach from that DNA molecule. So at this point, the mRNA molecule is going to be released as a free-floating single-strand copy of the gene. It's going to add a cap and a tail to this DNA, and then it's going to send it out of the nuclear pore in order to be copied. Now, when it caps and tails this for protection, it's also going to take the non-coding introns out and remove them. And so now all you have is the exons, a cap at the top, a cap at the bottom, and it leaves to go be copied. And so here is just another visual of what's going on. It recognizes a promoter site, goes in, starts copying, transcribes, terminates, caps the ends, removes the sections that it doesn't need, and sends it on out. So in summary, transcription is going to be the first step in a two-step process by which DNA directs the synthesis of proteins. In transcription, a single copy of one specific gene within the DNA is going to be made in the form of a molecule of mRNA, which moves where it can be translated into a protein. And remember, that's going to be at the cytoplasm. So translation, using information from DNA to build usable molecules. So several ingredients must be present in the cytoplasm for translation to occur. First, you have to have these free-floating amino acids floating around in the cytoplasm. Then you're going to also have to have ribosomal subunits. Remember, it comes in two parts. And so they're just floating around there in the pink or the red there. And then you're going to have what's referred to as a transfer RNA particle as well. And you're going to see that in the yellow particles there. So quick recap, ribosomal units, free-floating little amino acids floating around out there, and your tRNAs, which are going to go pick up those free-floating amino acids. Right, so looking at the specific tRNA, or what's referred to as the transfer RNA structure, it's going to actually, like we said, read the bases 3 at a time. When it reads them 3 at a time, those are referred to as codons. A moment to catch up. There we go. Those are codons. So this is its codon here. AUG is always going to be your start codon. That's going to be the promoter. That's where it says start here. And so it's going to attach to that and it's going to read those. It's actually going to create what's referred to as anti-codons here. So the anti-codons go out and search for a specific amino acid. It's going to pick that amino acid up, bring it back, and start the amino acid chain. So the attachment site is going to consist of a 3-base sequence that matches up with a 3-base sequence on the mRNA. Each 3-base sequence in the mRNA, we said, was called a codon, always matches with the tRNA that carries one particular amino acid back to the ribosome and starts the whole thing. So this molecule is very important in translation. So starting with translation, step 1 is going to recognize and initiate the protein. It always starts with the AUG, like I said. It's going to begin in the cytoplasm where the ribosomal subunits are going to come together. So the top subunit comes together with the bottom and the mRNA goes through the center. Reads that start sequence, which is always AUG, goes out, finds the amino acid, comes back, and starts the sequence. Here's going to be a chart of the codon table, which is going to show you which amino acid each of the 3 letters will code for. And so we said AUG is always going to be the start codon, and there's only one, so you always know to go with that AUG. And so you see that here. How you read this, you take the first letter, which is going to be on this side, and so we had A. The second letter is going to be up top, so we saw the U here, and then the third letter is the G. So we have A coming in, we have U coming in with the A's here, and then we find the G, which is going to be over on this side that can match up with them. And so it's going to be this one right here. Bring it on over. And there you have AUG, your start codon. Now to find any of the others, you would just use this chart, match up the letters the same way. So with step 2, we're going to do what's referred to as elongate or elongation. After the mRNA strand sequence, the next codon is going to be read, the correct amino acid is going to be brought to it by the tRNA, and then the ribosomes then facilitate that connection of this unit of second amino acid to the first. And like we said, they're going to hook it together using peptide bonds. Once you hook many of those together, poly means many, it becomes a polypeptide bond or a polypeptide chain. And so here's just a visual of what's going on with a different rendition from a different artist. And it shows you how each of those is fitting into, hold on, back. Give it a second to kick in. Okay. How you have these 2 ribosomal subunits that come together. They're going to have spaces for these tRNAs, which will read the codons, then they make like an anti-codon. They go out, they find a specific amino acid, bring it back to this ribosome, and then they start to hook it together to make that protein chain. So in summary, translation is going to be the second step in the 2-step process by which DNA directs the synthesis of proteins. In translation, the information from a gene that has been carried by the nucleotide sequence of mRNA is going to be read. The ingredients present in the cell cytoplasm are then going to be used to produce that protein chain. Genes are going to be regulated in several different ways, and we refer to this as gene expression and gene regulation. Microarrays are going to be a powerful tool that we're going to use to monitor the expression levels of thousands of genes simultaneously or at the same time. Particularly useful in exploring how gene expression differs in responses to things such as illness, treatment of illnesses. So people might have different genetic makeup, and because of their different genetic makeup, it might determine how they react to certain things, and or response to aging. So we're going to talk about how different makeups in the gene or in your DNA can actually change how you age, whether you're a fast ager or whether your aging process is going to be much slower than others. So looking here, we see lonely, isolated individuals are going to have very different patterns of immune system gene expression. They've actually found that people who are depressed or lonely tend to turn off genes that help with their immune system, or if they have a pretty healthy genome, it could actually inactivate some of that, and they could tend to get sicker more often and suffer from other things that normally they would not suffer from. So looking here, they had a 5-year medical study where they explored the health consequences of loneliness. They're going to use the microarray analysis of gene activity in immune system cells, and they're going to reveal that there's 209 genes that were expressed differently in 2 groups of participants. And this was just with this one study. So there could be more. All right, so 2 groups, so you have chronically isolated, and then you're going to have a normal group. Individuals in the lonely group had impaired transcription of some genes associated with the effective stress response, and they're also going to have an increased activity in genes associated with inflammatory processes that are going to be implicated in a variety of diseases. So a lot of different diseases or illnesses are simply inflammation of certain parts of the body that normally under normal circumstances or conditions would not be inflamed. So controlling gene expressions, you have your transcription factors. These are going to be proteins that bind to specific regulatory sites on DNA, usually located in the front of the gene. If it is a positive control, this is going to speed up that gene's expression. If it's a negative control, it's going to slow down or even block that gene's expression. So looking here, we have the Operon overview, so prokaryotic gene control and the Lac Operon. So with this Operon, it's going to be a group of several genes along with the elements that control their expression as a whole unit with all the sections of the DNA here. So the size of the elements are not going to be shown to scale, but the promoter and the operator regions are typically much shorter than the gene itself. And so here you have the promoter region, and so that's going to be the region of DNA that RNA polymerase recognizes that it needs to bind and start copying here. So it goes in, it sees it, and that's where it's going to bind. The region of DNA that a repressor protein can bind to is going to be the operator. And so by doing this, it's going to block the RNA polymerase from transcribing these genes. So it could go in, and if you have an operator here, it can stop it so it can't copy the gene that you need. Regulatory genes are going to be genes which can be located within the operon, near the operon, or even elsewhere in the genome, and codes the repressor protein that impedes transcription or activator proteins which enhance transcription. So there you see the operator. You see it can work with the protein repressor, or it could work with the activator. So elements of gene control. When you're going in with a promoter, this is going to be for a gene to be transcribed. The RNA polymerase has to recognize and bind to this promoter the specific sequence of nucleotides in the DNA that signal it to begin that gene. The operator is going to be a molecule called a repressor protein that can bind to the operator or the regulatory protein of DNA, and by doing so it can block RNA polymerase from transcribing genes necessary for lactose metabolism here. With the regulatory gene, it can code for the repressor protein that when bound to the operator region does the blocking of RNA polymerases that are binding to that protein site. So let's look at the Lac Operon in action. So if lactose is not present, you haven't eaten any ice cream or drinking any milk, so you don't have any lactose present. That repressor protein is going to bind to the operator. It prevents the RNA polymerase from binding to the promoter and transcribing the gene necessary for lactose metabolism because you don't need it. Lactose isn't present. However, if lactose is present, let's say you did eat the ice cream, then the operon will be on. So lactose, slightly modified, is going to bind to the repressor protein and it's going to prevent it from binding to the operator. Then the RNA polymerase can then bind to the promoter and transcribe the gene necessary for lactose metabolism. And so that's going to allow the gene to be made. Eukaryotic gene control. There's going to be many other ways that genes can be regulated besides these operons. Here you can see the different transcription regulation. You have what's referred to as activators and repressors for transcription. You have enhancer sequences, and you have chemical interference, and then you have post-transcription regulation. So let's look at the transcription regulation first. You might have positive controls, which are going to activate and initiate or speed up gene expression. You have what's referred to as enhancers, and what they're going to do is they're going to speed the RNA polymerase binding to and the gene transcription. If it's a negative control, repressors can block or slow down the gene expression, and chemicals can bind to DNA and actually block the gene transcription altogether. Now, if it's after transcription regulation, so post-transcription, that mRNA processing, transport, and enzymatic breakdown can be blocked or accelerated, or the mRNA translation and protein processing rates can be altered. So in summary, environmental signals influence the turning on and turning off of genes. By binding to DNA, regulatory proteins can block or facilitate the binding of RNA polymerase and subsequent transcription of genes. So damage to the genetic code is going to have a variety of causes and effects. So causes and effects of a mutation or a damage to DNA is basically an alteration of sequences of bases in DNA. They can lead to changes in the structure and function of proteins, and remember, structure is function. So if you change anything about how it's put together, lots of times it will code for something different, and lots of times it just doesn't even work. And so it could have a range of effects. It could actually even code for something worse or something that doesn't work correctly. Looking here, you have a normal fruit fly, but if you look over to the right-hand side, you have a mutant fruit fly. And so with a mutant fruit fly, it's going to have two small eyes in the place of antenna. It's going to have the large compound eyes right behind it. In humans, the breast cancer gene is going to be found within the DNA. So there's going to be two human genes called the BRCA1 and the BRCA2 gene. More than 200 different changes in the DNA sequence of these genes have been detected. And so they do look at this if they DNA test to see if you carry the specific genes that make you more susceptible to getting breast cancer. So each of which results in an increased risk if you have one of these two genes and there is a mutation within those 200 places that could be mutated. So mutations do have a bad reputation because lots of times we think of mutations as causing genetic problems such as breast cancer or prostate cancer and things such as that. But they don't always have to be bad. They tend to be what we refer to as disruptive. And lots of times mutations are actually very, very rare because we have a sequence that goes in and checks everything once it's put together. So mutation in sex cells and non-sex cells, is there a difference? Which one's going to get passed on to your offspring? Which one's just going to affect you? Well, the difference is, if it's in the sex cell, we're saying the sperm or the egg, then that mutation will be passed on to your offspring. If it is in a somatic cell, which is going to be a simple body cell, then it will not get passed on to your offspring if it's not in the egg or the sperm. That's only going to affect you. That is a mutation that occurred within your body, and it will not be passed on. But if it does make it to an egg or a sperm, then it can be passed on to your offspring. So types of mutations, you have what's referred to as a point mutation. These mutations are going to be where nucleotide base pairs in a DNA sequence is going to be replaced with another, in which the base is going to be, it could be inserted, deleted, or just simply replaced. Insertions and deletions can be much more harmful than a substitution because it can alter the reading of the entire sequence. So if it was ATTA, and you took out one of the T's and it just becomes ATA, then you're going to move the entire line of sequence that gets read down, and then that could create a whole problem because the entire gene is going to be coded differently, and so the gene would be off and it wouldn't work. Chromosomal aberrations are going to be changes to the overall organization of genes on a chromosome. They can involve the complete deletion of an entire section of DNA on a chromosome. They can move a gene from one part of a chromosome to another, so it could go from like a top to the bottom, or from the middle to the top or the bottom, so it just moves a whole section. Or it can duplicate a gene with a new copy and insert it somewhere else along that chromosome. And so here is a visual looking at the different types that we talked about. So with a point mutation, you're just taking away or adding one, or even just substituting one base. That can cause a complete change in sequence reading. Base pair insertion, where you go in and insert a whole section, or you can insert new ones. Or base pair deletions, you can delete a whole section. And here, looking at different ways to cause mutations, you will see them. So we have chemical causes with the smoking, UV radiation with everybody laying out in the sun, and you actually have things like X-rays, which can damage your DNA. So why is it dangerous around the core of a nuclear power plant? That's going to be because it's going to have a high chance of chemical-induced mutations there. So mutations are alterations in a single base or changes in a large segment of DNA that include several genes. They're rare, and when they do occur in a gene, it usually disrupts normal physiological functioning. They play an important role in evolution. It's how we change. So faulty genes and coding for faulty enzymes can actually lead to sickness. So how can people respond so differently to alcohol? Some people have specific genes that help them break alcohol down very quickly. Some people have specific genes that break it down at an average rate. And then some people are missing genes to even break alcohol down, so it becomes a toxic substance to their body. So a single difference in a single pair of bases in their DNA can actually make this change. One of these is referred to as a fast flush response. And so from mutation to illness, sometimes you don't even see unless you are exposed to something in your environment that causes you to see that there is a change in your DNA or something different. And so with this person, they might live a completely normal life. If they never encounter alcohol or don't drink alcoholic beverages, then they might not know that they don't have, or they have a faulty coding gene for this section of their DNA. So this is going to be aldehyde dehydrogenase. That is the enzyme that they're coding. And so this lady here, once she drank her 2 drinks, she started to turn red. And that's referred to as fast flush. That's going to be a buildup of toxins due to not being able to break down the alcoholic substance. So here with fast flush, we see some people who drink alcohol don't have the same effects of the substance as others. And so we saw the face redden. The heart's actually going to start to race. It produces nausea. And sometimes the respiration will start to increase. So they start to breathe a little heavier. And that causes a single base pair change. It makes that non-functioning enzyme, and it leads to actual physical illness. So alcohol breakdown in 2 process steps. So the first step is going to require the use of aldehyde dehydrogenase, which is an enzyme. Anything that ends in ASE or ACEs are enzymes. So without this enzyme, alcohol cannot be broken down for the second step. It becomes a poisonous substance that starts to accumulate and build up, which ends up causing illness to them. So if you look over to the side here, it shows you the enzyme. And so that enzyme is going to have a different shape when it's non-functioning to the shape of if it was functioning. Take this non-functioning shape down here, try to plug in the substance it's supposed to break down, and it doesn't fit. It's like a lock and key system. Since it doesn't fit, those molecules start to build up, and with a lot of molecules, you end up sick because you can't process them. So with that fast flush response, this is what you see happening. You go from being perfectly normal, happy, to your heart racing, inflammation in the face, it's turning red, and starting to feel sick and nauseous. Why do many Asians have unpleasant experiences associated with this alcohol consumption? The reason is because approximately 1/2 of people in Asia have this DNA change in their genes. So their genes code for a non-functional aldehyde dehydrogenase. And so that is going to be one of those things that we see in ethnic DNA or in DNA of certain areas. So in a study of non-alcoholics, they're going to find that a lot of people in Japan were fast flushers. So they tend to see less alcoholism due to such an unpleasant experience when drinking alcohol. So those people tend to stay away from it. So from mutation to illness in just 4 steps. First, you have a mutated gene that codes for a non-functioning protein, usually an enzyme. Then that non-functioning enzyme can't catalyze the reaction as it normally would, so it brings it all to a halt. Well, if it brings it all to a halt, the molecule which it was supposed to have reacted with is going to start to build up and accumulate. It's kind of like a blocked assembly line, so it just starts to fill up. And then that accumulation is going to cause sickness, or it could even cause death. So in summary, most genetic diseases result from individual mutations that cause a gene to produce a non-functioning enzyme, which in turn blocks the functioning of that metabolic pathway. This is where we're going to stop for today, and we will pick up with biotechnology on our next lesson.